bang yung mga biochemical test natin so that we have the single substrate utilization, pwedeng depende sa enzyme present. So we call that the enzyme test. We have the tolerance test. If you remember, this sa ating bacterial growth and cultivation. When we say tolerance is the ability of the different microorganisms to be able to uh, grow in extreme environments. So, to-tolerate nila whatever environment they are in. Then we have the inhibition test. When we talk about the inhibition test, remember your AST. Parang ganun din yun because you're trying to see which one would inhibit the growth of the microorganism and which one would not inhibit it. So, sabi natin yan, resistant or susceptible. Then we get into the common tests that are used for the Enterobacteriaceae. Diyan na yung mga TSI, yung ating mga LIA, yung ating mga oxidase, and all of those and the others that can all lead to the identification of the organism. So we'll start with the single substrate utilization. So sabi natin, so that when we say single substrate, ano ba ang substrate? Diba alam natin na ito yung mga nag-act sa enzyme para magkaroon na dun sa chemical reactions either in the splitting or it can hasten the reaction. So the first we have two single substrate utilization tests. We have the acetamide and the acetate. If you went over your notes and then also you had the chance to get over the modules sa laboratory, then you will see that both tests rely on the use of carbon from different sources. So for acetamide, ano ginagamit natin dito is the acetamide. Dun sa acetate is sodium acetate. So that acetamide utilization, this is performed for the identification of aerobic organisms and it is limited only to few organisms kasi only few organisms are able to use acetamide as the sole source of carbon. And this is commonly performed for the differentiation of our Pseudomonas aeruginosa from other non-glucose fermenting gram-negative rods. So, ang media na ginagamit natin dito, acetamide agar slant. Also, although you are seeing in the picture, ang ating tube media ay naka-bat slant. So, hindi in-stab yung bat. Okay, we're not, ang ginamit lang dyan, kasi ang nakalagay lang dyan is bat slant, but what we're interested in is the slant only. So that the media na ginagamit dyan is our acetamide agar slant. Ang composition, ang components ng acetamide agar slant is, andun yung acetamide as the sole source of carbon, meron siyang inorganic ammonium salt, which serve as the sole source of nitrogen, and then, meron tayong indicator because it's the indicator na will be responsible for the change in color depende kung ano na yung end product of metabolism, whether it will make the medium now acidic or alkaline. So, ang indicator natin with both acetamide and acetate utilization is our bromthymol blue, and with acetamide utilization test, so, sabi nga natin dito, it is based on the utilization of acetamide by the organism via the process of the amidation in the presence of the enzyme. Ano yung enzyme natin dito? What is our enzyme when we're talking after acetamide utilization? Anyone? Yes, acrylamidase. So that by the amination in the presence of acrylamidase and resulting in the release of ammonia, which now will change the pH of the media into alkaline. Nag-i-increase ang alkalinity niya because of the release of ammonia. So, it will change now yung ating acetamide agar slant from green to blue. Of note or of emphasis here is the growth of the microorganism. So, pag nag-check tayo ng acetamide utilization or yung mga the use of carbon, acetamide as a sole source of carbon or whatever, importante na nag-grow si microorganism. Bakit kailangan may growth? Bakit kailangan may growth? Bakit kailangan may growth? Di ba pwedeng nag-change lang yung ating color, okay na yan, positive na yan, nag-change nga yung color eh. So, alam natin na naging alkaline yung ating acetamide agar slant. Bakit kailangan may growth? At anyone? Bakit kailangan may growth? We're talking of utilization. Kung walang growth, sinong gumamit ng acetamide? Bakit nagkaroon ng change in color? So that kahit nag-change yan ng color ng blue na walang growth, hindi nyo pwedeng sabihin positive yan. So that you always have to check. This will be true for your acetate utilization, also for your citrate utilization test. You always have to check for the growth of the microorganism plus the change in color from green to blue. Okay? So you have there your positive and negative control. Gawa na lang kayo later ng inyong tabulated form, mga positive sa ganitong test para at least then you can come up with your own summary. Okay, so that is for acetamide utilization. Now, the other single substrate utilization test here is our acetate utilization. So, pag acetate naman, kung kanina ang ginamit natin ay acetamide na sole source of carbon, or acetate in the form of sodium acetate, it is now the sole source of carbon. And, of course, here we see that the practical application differentiate from Shigella while most of the utilize. So the state by, of course, now is followed by the breakdown again of ammonium salts into ammonia. Kaya ganun din ang result niya. Pareho lang sila ng principle niya. It's just the straight is different. So, ang ating incubation dito, sabi ko nga sa inyo kanina, incubation acetate utilization is up to 7 days. So that lamang na lamang ang ating mga molecular techniques when we're talking of such things as masyadong mahaba ang ating incubation period. So again, you have there your positive control is of course your E. coli. Although some strains use acetate very slowly or not at all, and then ito magbibigay sa atin ng false negative. And of course, sabi nga natin, it's a shaping factor with Escherichia or Shigella, which does not utilize acetate as their sole source of carbon. So, dalawa lang yung ating substrate utilization. Diyan, you have your acetamide and then you have your acetate. Okay? Pareho lang ang result nila from green to blue. But again, sabi ko nga kanina, it's always important that you have to check that yung change of color was brought about by the utilization of the substrate because of the presence of the organism. So, we proceed with the different enzyme tests. So, ano na naman? Sabi natin mga enzymes. Di ba sabi natin kanina dun sa substrates? Yung substrates, these are the substances upon which an enzyme acts in a chemical reaction. So, what are the different enzymes? If you remember, you go back again to your lessons, di ba sinasabi natin, ano yung resistance ng microbe to the immune system of the host? Di ba they're able also to produce enzymes which protects them and which makes them able to survive in the environment. So, we start with our butyrate dish. Ano ang butyrate dish test natin? So that this is a rapid qualitative test for the detection of the enzyme butyrate esterase, which further provides a presumptive identification tool for our Moracella catarrhalis, which used to be Branhamella, and the hydrolysis of bromochloroindolyl substrate by butyrate esterase releases indoxyl, which in the presence of oxygen forms now our indigo. Okay, now indigo is a chromogenic compound that appears blue to blue-violet. So, i-review natin ngayon, we're talking of enzyme test. So, anong enzyme test natin sa butyrate this test is our butyrate esterase. And our substrate here is our bromochloroindolyl, which is impregnated in the disc. So, nakikita nyo mga disc na yan, parang sa antibiotic disc natin. So, you have, this one is impregnated with the bromochloroindolyl. Okay, so that, again, substrate, sabi nga, hydrolysis will give us the indoxyl. In the presence of oxygen, will give us now the indigo. Yun ang responsible for the blue to blue-violet color. But you see here that it should be within 5 minutes so that if nag-exceed tayo, we read our disk after 5 minutes that will be of positive reaction. Kasi usually within that 5 minutes, baka 3 minutes, mag-blue na rin yung ating disk. Okay? Now, when we're using naman two small inoculums, if not able to change or not able to tawag natin doon sa hasten the reaction, kukunti lang si microorganism, kaya magkakaroon din tayo ng false negative. So that if we are sure again, we get back our gram stain and then we get back our colony morphology and somehow medyo hindi tayo solve dun sa result ni butyrate this, we can always repeat the butyrate this test using now a larger inoculum. Okay, so again, we have our positive and negative control. Positive is our Morax lactosaralis, which we are trying really to identify with the use of this test. And negative is Neisseria here with our catalase test. Alam na natin, ano ba si catalase? So, saan yung unang narinig si catalase? And change breakdown of hydrogen peroxide into water and oxygen. When we were talking about what? Your anaerobes or your aerobes, diba? So, we all know now that catalase is an essential enzyme in pathogenic organisms. Bakit? It protects the organism from oxidative damage from reactive oxygen species. So that the catalase enzyme now neutralizes the bactericidal effects of hydrogen peroxide, and its concentration in the bacteria now has been correlated with the pathogenicity of the organism. Again, you see here the reaction. Pag meron kayong slide, nilagyan natin yan ng ating inoculum. Kumuha tayo na sa pinag-isolatean natin ng ating organism. So you just get a portion, you just touch that colony and then ilagay natin siya sa slide. Then our reagent here is 3% hydrogen peroxide. So ipro-provide natin sa hydrogen peroxide na dapat nare-release katawan, di ba? Para ano? We'll just see whether the presence of the enzyme catalase in the organism para makita natin na mabe-breakdown ng ating H2O2 to water and oxygen, and that water and oxygen, or is the oxygen now, that's as the bubbling, or what we usually say, the more specific is effervescence or copious bubbling, yung mga ganun na results sa ating catalase test. So that, again, of course, the practical application is the differentiation of the catalase positive organism like micrococci and staphylococci from catalase negative organisms like our streptococci. So that pag nakita niyo yung algorithm, ang gram-positive cocci, ano yung pinakaunang test doon would be a catalase test. Only catalase test to differentiate now your staphylococci from your streptococci. So doon ka na ulit mag-i-start. Gram-positive cocci ka, kasi di ba sabi ko nga, pag nag-gram stain ka, makikita mo na ang gram staining reaction no isolate mo, and of course the shape, so that and the arrangement. Kaya tayo merong gram-positive cocci in cluster or gram-negative bacilli singly or in chains. So that doon mag-i-start ang inyong algorithm. Pag nakita mong gram-positive yan, Staphylococcus yan. Gram-negative yan, Streptococci yan. So, mag-iisip ka na ngayon ng susunod na biochemical test mo para sa Staphylococcus lang. So, aside from that, your catalase test also differentiates aerotolerant strains of Clostridium. They are catalase negative from the Bacillus, which are catalase positive. It also differentiates Listeria, positive again for catalase, from erysipelotrix. These are catalase negative. And even the anaerobe Bacteroides is also catalase positive. Sabi nga natin usually ano ito sa mga aerobic. But Bacteroides is anaerobe, but it is catalase positive. Now, sabi ko kanina sa inyo, ang ating reagent dito with the catalase test, when you're checking on for the staphylococcus, for differentiation of staph and strep, ang ating reagent ay 3% hydrogen peroxide. But, pag nakita nyo na ang reagent ay 30% hydrogen peroxide. This is used in other biochemical tests like salt test. Kaya siya may super doon sa umpisa niya. This is screening of Neisseria gonorrhea. And all for the, we can also test for a heat stable catalyst, which is present in, which is a biochemical test for our mycobacterium species. Okay. Please also take note that when we're doing the catalyst test, hindi tayo pwedeng mag-pick ng colony of a blood agar plate. Remember, we're trying to identify staph from strep, and we can inoculate or we can do our subculturing on a blood agar plate kasi nga di pa naman natin alam kung ano talaga yun. So, bakit hindi natin pwedeng gamitin blood agar plate? Di ba pag gumawa tayo ng BAP, ano ang enrichment natin sa BAP? Anong nilalagay natin na enrichment ng ating BAP? Chat box or kung map, mic, pwede naman. Ang enrichment ang nilalagay natin sa ating blood agar plate or our BAP. Enrichment.
Ma'am, yung 5% sheep agar po.
Okay. Yes. Okay. So,
Sheep
di ba
blood.
tinalagay natin ng blood
True.
to blood agar? So, pwede siyang sheep blood. Kung meron, dapat sheep blood talaga. Pero kung walang sheep blood, pwede namang human blood. Defibrinated human blood. Okay? So that, ano ang meron si blood na magiging parang catalyst din siya? Ano meron si blood? Anong composition niya? Na kaya nagkakaroon tayo ng hemolysis. Pag yan ay may presence na hemolysis. Ano meron si blood? Siya ay may na ginagaya kaya pseudoperoxidase activity na nare-release pag nag-lyse si RBC.
Hemoglobin, ma'am.
Hematology. Hemoglobin.
Him, ma'am.
Okay. So, blood. Okay, pareho na. So, that, yun yung hemoglobin, meron siyang pseudoperoxidase activity. Ano yung pseudoperoxidase activity? Kinakatalyze din niya the same reaction na parang ginagawa ni catalase enzyme. Kaya ba ginagawa ni catalase enzyme? Di ba breakdown niya sa hydrogen peroxide para mawala yung toxicity ng hydrogen peroxide din sa cell. Okay? So that, ang positive control natin dito is when we're talking of catalyst, anong positive control natin? Kahit hindi nyo tinitignan, kunyari, yung ating slide. Diba sabi ko kanina, differentiating your gram-positive cocci. Your positive control would be your Staphylococcus aureus. Negative control natin would be Streptococcus pyogenes. Okay, so dalawa na ang enzyme nyo. Anong enzyme natin can butyrate this test?
Butyrate
Para at least
esterase.
paulit-ulit tayo. Butyrate esterase. Kay catalase. Tagal.
Catalase
Catalase.
enzyme.
Kaya nga, catalase. Kaya nga sabi ko, ang tagal. Catalase enzyme. Now we get into the next. Perform ng clot. Ano yun? Okay? We get into our coagulase test now. So coagulase, again, is another enzyme. Okay? So that it is an enzymatic protein that is a thermostable thrombin-like substance which converts fibrinogen into fibrin, resulting in clotting or clumping. So yung coagulase enzyme also acts as a virulence factor in some organisms. Why? It interacts with the fibrinogen present on the host cell surface. So that organisms with coagulase usually have a protective barrier around themselves, increasing now their pathogenicity and resistance against the immune system. So, ang practical application na naman natin dito, you always get your practical application doon sa inyong self-study guide, is that it differentiates now the genus Staphylococci into two groups. Ano yung two groups? Now, you have your coagulase positive and you have your coagulase negative. So you have your CONS or CONS, C small o and S, that is your coagulase negative staphylococci. So that alam natin na ngayon, doon sa ating catalase test, nag-positive ng catalase, alam natin staphylococcus yung next na gagawin natin, coagulase. Bakit? Kailangan natin malaman kung sino ba si positive at sino si negative. Masa-separate na natin yung mga medically important staphylococcus natin. So that our coagulase positive would be our staphylococcus aureus, and our coagulase negative would be our staphylococcus epidermidis and saprophyticus. Okay, now coagulase is of two types. Ano yung types natin ng coagulase and differentiate one from the other? Anyone? Two types of coagulase. Anyone? Just a slide. Just differentiate one from the other. Okay? Sabi ni Ria, bound and free. Sabi ni Brandon, bound and free. Okay, differentiate one from the other lang, Brandon. Ano si bound at ano si free?
Ma'am, yung bound po is nasa cell wall po. Tapos yung free is hindi po siya naka-attach dun sa cell wall. Ganun po yung pagkaintindi ko, ma'am.
Tama yun. So what test now? Bakit, anong test ang gagamitin mo kay bound? Kasi aside from the classified as bound and the free, so yun din ang nagsasabi sa atin kung ano yung test for that, for coagulase. So pag sinabi natin bound, yun yung sinabi natin bound.
Slide
we also
test
call
po,
it
ma'am.
slide test. Yes, slide test for the bound. So that it is also called the clamping factor. Diba? Kaya nga, slide test ang ginagamit natin. Sabi nga ni Brandon, naka-attach siya sa cell wall. And then this one now, being attached to the cell wall, it reacts directly with fibrinogen. Okay? So that you can see now your positive results within 10 seconds. Now, what happens with our free coagulase? Pag nag-negative si slide test, isasabihin nyo na ba na negative coag yan? Do you stop at the slide test? Wilson, do we stop at the slide test? Diba dalawa sabi natin, bound and the free. The bound is the slide test, the free is the tube test. O nag-slide test na ako, di ba, kumuha ka ng inoculum, nilagyan mo siya ng rabbit plasma ulit, and then wala kang nakitang clot formation. So do you stop there? Do you tell now, do you give me now a reason? Ma'am ko, kung nasa lab tayo, ma'am ko, wag negative po. Walang clot formation sa slide. Wilson. Asan na si Wilson? Pwede lang ulit sa chat box. Ria.
Yes, ma'am.
Ria?
Po.
Okay. So slide test, di ba sabi natin bound and free, si bound ginagawa ng slide test. So that if nag-negative tayo sa slide test, anong gagawin mo? You stop there? Coag neg na yun?
Ma'am, magpro-proceed po ata sa tube test.
Yes, you proceed. Proceed ka sa tube test. Bakit? Baka hindi nga bound, hindi bound coagulase ang, kasi sabi natin, di ba, dalawa ang pwedeng forms ng coagulase. So, pwedeng hindi siya bound at ang meron siya free. So, you proceed with the tube test. So, how do you do your tube test? Ano? Tatayo muna ako. Kanina pa ako nakaupo. Paano yung sa tube test natin? Sabi natin, it detects the free coagulase. So that our free coagulase is different from bound coagulase in that, kaya nga meron tayo dyan, extracellular protein enzyme. So that the clotting mechanism of our free coagulase now requires the activation of a plasma coagulase reacting factor. Yun yung ating CRF, which is a modified or derived thrombin molecule to form a coagulase CRF complex. Ito ngayong complex na ito ang magre-react with fibrinogen to form the fibrin clot in a test tube. So that ang pagtingin nyo yan ng fibrin clot, kung minsan parang nag-solidify, parang gelatin din na gumagalaw, pero hindi na siya liquid talaga. So the only thing with the tube test is that, so you put now your reagent. Anong reagent natin sa tube test? That's our reagent. Anong reagent natin sa coagulase test, sa tube, kahit sa coagulase test, anong reagent natin? Okay, rabbit EDTA plasma. So that plasma now, you're going to put 0.5 ml, 0.5 ml is for 4 to 6 hours or sometimes overnight. The thing there is, kailangan natin siyang tignan after 4 hours. Bakit natin kailangan siyang tignan after 4 hours? If there is no clot formation kasi after hours, you can proceed to incubating it overnight or siguro 6 hours. Some books say 4 to 6, some say overnight. So if we're going to proceed with overnight incubation, kailangan natin siyang i-check every 30 minutes. Bakit? Bakit kailangan natin siyang i-check every 30 minutes? Kasi yung ating pwede siyang mag-form ng clot ewing and then it can revert back. So, pag hindi natin kita yung clot formation na yun, then we will be saying again that it is coagulase negative. Okay, so the incubation is 35 to 37 centigrade. And there is a question in your study guide whether you can use EDTA anticoagulant, rabbit EDTA nga kianin. Hindi, citrate pala yung hindi pwede. Okay, you can use your EDTA anticoagulant, but you cannot use citrated plasma. Why? Bakit hindi pwede si citrated plasma? Why can't we substitute EDTA with citrate? Bakit? They can sa notion themselves ito. Some organisms are citrate utilizing. Nag-lyse po siya. It prevents clotting. Eventual. Therefore, also may cause, hindi siya, kasi magpo-cause siya ng false positive result so that marin siyang clotting. Okay, because of the utilization now of citrate. So, we have there our positive results na natin using the principle of coagulase test, clumping or clotting of. So, that sometimes kasi sa slide, pwede rin natin i-view yan sa microscope pag-li. Yung just to walang clumping. So that our positive control would be our Staphylococcus aureus, which is coagulative, and of course, Staphylococcus epidermidis and or Staphylococcus subtypicus, which are our coagulase negative Staphylococcus. Okay. Okay pa kayo. Ilang test pa lang yun. Isang katerba pa ang kasunod niya. Diba? Sabi nyo nga, overwhelming. Sobra talaga. So we proceed to the other enzyme test, our DNA hydrolysis. So ano si DNA hydrolysis naman natin? So we know now, di ba sabi natin kanina, meron tayo dun sa first part, dun sa ating, when we're trying to also do molecular testing. We have the genotype of the organism. So in here, DNases are enzymes that hydrolyze our DNA and release free nucleotides and phosphates. So the deoxyribonuclease enzyme, yun ang ating enzyme dito or our DNase, is produced by bacteria. This is extracellular endonucleases that break down our DNA, yielding now a high concentration of oligonucleotides. And the media used to detect these enzymes can be made by using various indicators. Pwedeng may indicator, and indicator could be toluidine blue or methyl green, or pwede ring walang indicator to detect now the hydrolysis of DNA. Yung method dito natin sa Denise test agar is wala tayong indicator dito. So what happens? The hydrolysis of the DNA is indicated by the clearing of the agar after the addition of HCl, so that nagdi-dissolve the oligonucleotides dissolve in acid, causing now a clear zone. But DNA salts are insoluble. So, you see here now, yung clearing dyan is already because of hydrolysis of DNA. Okay, so aside from that, Serratia species positive from Enterobacter and all, DNA hydrolysis is also used to differentiate. Andiyan na pala. You have Serratia species, and then your Cataralis, and of course, your Staphylococcus aureus. So if our methyl green here is uninoculated, yung ating DNA methyl green media will still will remain green. Pag na-hydrolyze si DNAs, then magkakaroon ng colorless. Yun yung clearing sa streaking, kung saan natin na-streak yung ating organism. So that, sabi nga kanina, we can use different, pwede tayong gumamit ng indicator. Pag ang indicator natin ay toluidine blue, so the toluidine blue plus the DNA, again, will have not a colorless result now, but a blue result. When DNase hydrolyzes this complex, toluidine is reduced and converts to rose pink. Okay? So positive result for the methyl green is clearing around the colonies. Okay, positive control natin, Staphylococcus aureus. Negative control natin ay Staphylococcus epidermidis. Okay, so pag walang indicator sa DNA hydrolysis, how can you see whether there was hydrolysis of DNA? What would be the positive result? No indicator. Positive result, no indicator.
Clearing po, ma'am.
May indicator, for example, it's toluidine blue. Pag nag-toluidine blue tayo, kasi pwede ka mag-indicator ng toluidine blue or methyl green. So that, pag toluidine blue, the toluidine with DNA,
Toluidine blue
will
po,
be
blue
blue.
result.
Magbo-blue muna siya, but when the DNA enzyme or your DNase, deoxyribonuclease enzyme, hydrolyzes your toluidine blue plus DNA, which is blue. The toluidine is reduced and converts to rose pink. Okay? So, yung DNA mo plus yung toluidine blue na indicator mo will be colored blue. Present si enzyme, your DNase or your deoxyribonuclease enzyme, it will now hydrolyze that complex, the blue complex, toluidine plus your DNA, and toluidine is reduced and converts to rose pink. Okay? So, pag yung sa methyl green, colorless. Pag sa toluidine blue, ang end result mo would be rose pink. Yung complex na toluidine blue plus your DNA will be what is colored blue. Gets? So, from blue to rose pink. Si methyl green is from methyl green na green to colorless. Okay? Clear tayo kay DNA hydrolysis. So, we proceed now to your gelatin. Okay yon? Gelatin hydrolysis. Ano naman si gelatin? Ano ba si gelatin? Di ba, very familiar naman kayo with the gelatin. And we even try to do that with our homemade culture media. So that here, gelatin hydrolysis test is used to detect the ability of an organism to produce, again, enzyme-based tayo. So it's the enzyme gelatinase, and your enzyme gelatinase is a proteolytic enzyme. When we say proteolytic enzyme, anong ginagawa nila? Yung ating mga proteinase or yung mga protease, ganun. Anong ginagawa nila? What is a proteolytic enzyme? So that they break the long chain like molecules of proteins into shorter fragments. Ano yung shorter fragments? Hanggang maging amino acid yata sila. So this process takes place into sequential reactions. Ano yung dalawang reaction? Doon sa unang reaction, gelatinase degrade gelatin to polypeptides. Okay, after that, the polypeptides are further converted into amino acids. Now, the bacterial cells can then take up this amino acid and use them in their metabolic processes. So, dito ngayon, kasi ang ating positive result, diba, sabi natin dito is total or partial liquefaction. If you observe when you bring out gelatin at hindi tama yung mixture, hindi pwede mag-liquefy yan. So that in here, pag nag-inoculate na tayo ng heavy inoculum ng test bacteria, stab siya dun sa bat ang ating, ano dito ha, bat yung ating gelatin, dun sa tube. So that pag bat ang ating media, ang manner of inoculation is by stabbing. Okay? So that after you have stabbed the organism, the test organism into our agar, nutrient gelatin agar. Pinalitan natin si agarose into gelatin. Yun ang ating media. Incubate siya, both inoculated and non-inoculated or the uninoculated. Why? Because we would like to check later on kung yung liquefaction ba ng ating nutrient gelatin agar was brought about by the presence of the organism with the enzyme gelatinase. Kasi sabi ko nga, di ba gelatin yan, pwede naman yan mag-liquefy kahit walang organism. So that magkakaroon ka ngayon ng false results. So that you would like to have one that is with the test organism and one that is without the test organism. And then dito, you have to remove the tubes daily from the incubator and then ilalagay mo siya sa refrigerator para tignan lang kung babalik yung ating control, yung walang inoculated na Siyempre, pag tinanggal mo sa incubator, iba ang temperature. Mag-aalala yung mga temperatures natin, magli-liquefy siya. So, yun ang pang-check natin. Okay? So that you tilt the tubes to observe if gelatin has been hydrolyzed. So that gelatinase is present, then the gelatin will liquefy. So that we have here positive result, of course, is partial or total liquefaction at 4 degrees centigrade within 14 days. Again, negative, of course, will be complete solidification of the nutrient gelatin agar. Okay, so our positive control is Bacillus subtilis, or we can also use our Serratia marcescens as a positive control. And negative control, it can be E. coli or also Enterobacter aerogenes. Okay? So, gelatin hydrolysis, ano ang ating enzyme?
Gelatinase.
Gelatinase.
Gelatinase
Saan?
po.
Teka lang, may nakita ako bigla. Israel, saan yung bronze color? Rose pink, malapit ba siya sa ano?
Ay, ma'am,
Israel.
misheard ko lang, ma'am. Rose pala yun.
Rose pink, from blue, pag toluidine, rose pink, from blue to rose pink. Hindi, bigla ako napatingin lang. Baka mamay, asan yung bronze color? So, it's rose pink to, blue to rose pink. Yung complex mo, DNA plus toluidine will give you the blue color. Hydrolysis now, in the presence of the enzyme, will now be hydrolyzing toluidine now into rose pink. Okay? So we proceed. Gelatinase, you have your gelatinase. So if the enzyme gelatinase is present in the organism like Bacillus subtilis, then ano makikita niyong positive result would be partial or total liquefaction at 4 degrees centigrade within 14 days. Okay. Okay pa kayo? Ilan pa lang yun sa
Yes
enzymes
po, ma'am.
natin. Okay. So, gelatinase, anong result natin?
Partial or total
Result lang.
liquefaction, ma'am.
Liquefaction.
DNA hydrolysis. Methyl green, anong result natin?
Colorless
Colorless.
or clearing, ma'am, of the green zone.
Oh, dapat mas maganda yung clear, hindi lang pwedeng colorless ha, dapat kasama yung clearing around kasi yun yung magbibigay dun sa ano. Pag sinabi natin colorless lahat yan, nawala lahat ng color ni methyl green dun sa ating media around the colony. Kasi doon mo in-strict yung microorganism mo. Okay? So that, how about our coagulase test? Coagulase.
Depende sa type of coagulase.
Ay, hindi, pareho lang result nila kahit siya ay free or bound.
Ma'am, presence of clot
Di
po.
ba?
May clot.
Oh,
May clot
o
po.
yung clotting, pareho lang, hindi pwedeng iba ang result ni free at iba ang result ni bound. Kasi you're checking for the clotting mechanism pa din ni coagulase enzyme mo. Catalase, anong result? Ang tagal.
Bubbling.
Bubbling.
Bubbling. Oh,
Bubbling.
bubbling churva. Walang char, ganun. Bubbling. O, ano ang
Ma'am,
reagent
mayabang
ni
si
catalase?
Dex, ma'am.
Bakit? Bakit mayabang si Dex? Dex, mayabang ka daw, oh.
Ma'am, inaaway niya ako.
Alabad ka, Dex. Huwag kang mang-away, Dex. May message si Dex. Hindi, ma'am daw. Okay, catalase reagent. A-answer daw si Ma'am
Catalase po.
Arlene. Reagent. Ang tagal, bilisan nyo H2O2, sabi ni Kevin. Okay, very good, Kevin. Coagulase. Reagent. Ma'am Arlene, coagulase. Coagulase. Ang tagal.
EDDA, ma'am.
Dexter. Ano Dexter? Andyan si Dexter. Edta ano? Ay, pogi daw, Dexter, sabi ni Ma'am Arlene. Rabbit edta ano, Madam ba? Rangel? Kulang naman. Rabbit ed. Ay, very good. Ay, very good. Dati ko na. Dave, hindi pwede frozen plasma. Hindi pwede frozen. Hi, ma'am, ganda daw, sabi ni ma'am ni Dexter. Wow ha daw, Dexter. O, okay na kayo, gising na kayo hanggang alas 5 tayo, matapos natin ang biochemical. Ako gigising sa inyo. O, game. Okay? Bilisan nyo naman yung ano, pang ano na natin to. DNA hydrolysis. Anong reagent? Anong reagent? Tignan ko. O, DNA hydrolysis.
Methyl ritual within blue.
Ha? Ano? Ulit, please. Nasa enzymes tayo. Anong ating ano? DNA hydrolysis. Pagkatapos natin, mamaya, gumawa kayo ng concept. Okay. The presence of your deoxyribonuclease enzyme. Indicator natin sa DNA hydrolysis.
Methyl
Methyl
green
green.
toluidine blue.
Yes, methyl green? Pwedeng toluidine blue. Okay, very good. Kathleen, Vincent. O, gelatin hydrolysis. Tanggalin ko na Ano nangyari sa ating gelatin hydrolysis? By daw, Dexter, sabi ni Ma'am Arlene. Ha? Anong media natin sa gelatin hydrolysis? Media. Methyl red si ano. Nutrient gelatin medium. Thank you, Lorraine. Okay, so we proceed. Okay na kayo. Punta naman tayo sa ating hipurate test or our hipurate hydrolysis test. So, our hipurate test is based on the ability of the organism to hydrolyze again your sodium hipurate to benzoic acid and glycine by the action of the enzyme. Yung enzyme?
Hipuricase.
Enzyme. Hipuricase. Okay, so that the rapid test involves ninhydrin as the indicator. The ninhydrin will react with any protein or amino acid. Dito, ang nag-react siya with, of course, our glycine. Now, the glycine is deaminated by the oxidizing action of ninhydrin, which causes the reduction of ninhydrin, resulting now in a purple colored complex or a purple colored product. So the test medium used here must only have hipurate as a protein source since ninhydrin reacts with any free amino acids present. So we have also the rapid hydrolysis test, which this is more specific. It detects also the benzoic acid byproduct and has been shown to be as sensitive and as specific still with this hipurate hydrolysis wherein our end product here is only glycine plus benzoic acid. Okay, so enzyme is enzyme. Yung
Hipuricase.
enzyme. Anong oxidizing agent natin?
Ninhydrin po.
Hipurate. Yes. Oxidizing agent is ninhydrin, which will lead into
Purple color. Ay, deamination of glycine po.
deamination of glycine, which now gives us our purple
Purple colored
colored
product.
product. Okay. Okay, so purple colored. Okay, so you have again, positive control is Streptococcus agalactiae. Your negative control is Streptococcus pyogenes. Proceeding into our leucine aminopeptidase test or the lab test, which is of course now a rapid test for the detection of the enzyme leucine aminopeptidase or our lab. This is a presumptive identification of the catalyst negative gram positive cocci. Ano si mga catalyst positive gram positive catalyst negative gram positive cocci? Doon sa ating test kanina, pag nag-catalase test tayo, sino si catalase negative gram positive cocci? Sino yung mga yan? Mga gram positive cocci, you do a catalase test. Gram positive would be asan na?
Ano, ma'am?
Unang ano pa lang yung unang algorithm. Ano?
Ma'am, catalase positive yung staphylococcus.
Okay.
Catalase negative
Yung
streptococcus.
catalase negative.
Streptococcus.
Okay, so this, streptococcus. So that, ang ating leucine beta-naphthalamide impregnated disc Although what is shown here is yung naka-cotton swab siya. So kumuha yan. Meron ng dis yan, parang yung sa oxidase test din. Ganun, or sa butyrate dis test. Parang may mga dis din sila. And then that dis is impregnated with the leucine beta-naphthalamide, which acts as a substrate for the detection of the enzyme leucine aminopeptidase. So that the enzyme leucine aminopeptidase now hydrolyzes the substrate, resulting in the production of beta-naphthylamine, which upon the addition of your para-dimethyl aminocinnamaldehyde reagent form a highly visible red color shift base. Yung mga reagents na yan, kailangan ninyo lahat i-memorize yan, gaano man sila kahaba. Okay? So, the practical application here, of course, will be to detect the presence of the enzyme leucine aminopeptidase for the preliminary characterization of your catalase negative, gram-positive cocci, especially the non-beta hemolytic cocci. If you take a look at this slide, sabi dyan dapat again is the development of a red color within 1 minute. Again, the incubation time there is very important, as with all the other enzyme tests. So dito ngayon, negative result natin, of course, there will be no color change or pwede rin siyang slight yellow color. Next, ano ang ating enzyme sa lab test? Enzyme
Leucine aminopeptidase po.
Okay, thank you. We proceed. Microdase test or the modified oxidase test. Sino na yun? Si Dexter? Si Brandon? Brandon,
Po,
Brandon, explain mo daw yung microdase modified oxidase test, sabi ni Ma'am Arlene.
wait lang
Ba't ka
po.
natatawa, sabi ni Ma'am Arlene? Nakikinig si Ma'am Arlene.
Microdase test differentiates Staphylococcus from Micrococcus species by detection of the enzyme oxidase. So, yung oxidase daw, ma'am, reacts with the oxidase reagent and cytochrome C to form the indophenols. So, yung indophenol po, ma'am, is yun po yung nagkakos po nung blue to purple blue color, ma'am. Kapag positive po siya.
Ano ang oxidase reagent natin? Oxidase reagent.
Hindi ko po alam, ma'am.
Hindi nyo ginawa ang self-study guide ninyo. Ano ang oxidase reagent natin? Okay, tama ka dun na. Okay, differentiate. Staphylococcus from Micrococcus, which are gram-positive cocci possessing catalase enzyme ulit. Anong oxidase reagent natin? Brandon. Brandon daw. O meron tayo dyan yung ating tetramethyl paraphenylene diamine dihydrochloride indimso or our dimethyl sulfoxide. Okay? So that the modified oxidase reagent is prepared with 1% weight over volume tetramethyl paraphenyl diamine in certified with dimethyl sulfoxide. Ano ang role ni dimethyl sulfoxide? It aids in the permeability of cells to the reagent. So that in the presence of atmospheric oxygen, the oxidase reagent, the oxidase enzyme reacts with the oxidase reagent and cytochrome C. You'll always see, basta meron tayong oxidase test, oxidase something, like ano pa, spot test or whatever. The cytochrome C to form the indophenol or the colored compound na blue to purple blue color on the disc. Nakita niyo yung picture ng ating microdays after the introduction of bacterial colony on the disc. Okay? So, ilan na yung i-memorize nyo na reagent? Balikan nyo mamaya, ha? Okay? Nahilo na. Ilista nyo isa-isa. Next! Hindi, pag nagpa-quiz ako, hindi ko pagsabay-sabayin para ma-master nyo dapat. Mastery ang ating targeting dito. Okay, kahit araw-araw tayo mag-quiz. Lucid, alina peptidase.
Rant ka dyan.
Hindi, mabait ang Tracy, hindi ako irarant.
Irarant ka ni Dexter.
Dexter, irarant mo ako. Hindi na nga makarant si Dexter. si Ma'am Arlene ang nagrarant kay Dexter ngayon. Okay, next. Iba naman. Si Ma'am Arlene lang po. Ang irarant mo?
Sige, magrantan tayo. Tingnan natin. Open ako ng Twitter para sa'yo.
Gagawa daw siyang Twitter account, Dexter. Millennia. Ha ka daw, Arlene. Millennia. Paki naman sa mag test. May kaparehong test ito, bago lang natin na
Kape test yan.
fluorescence lang ito. Yung isa kanina, iba ang result. Pero practically pareho ang kanilang principle. Millennia. Asan na si Millennia?
Ma'am.
Sa'yo naman si methyl ambaferyl kasi mabulol-bulol ako kay ambaferyl beta D-glucuronide test or the MAG test.
MAG test is used
Yeah.
to presumptively identify various genera of Enterobacteriaceae and their toxin-producing Escherichia coli.
Ano ang role ni E. coli? Bakit ano yan? Ano ang role ni E. coli dyan sa kasi mag lang ginagamit natin sa kape, hindi pang biochemical. Dito na ako magkaklase lagi kasi andito si Ma'am Arlene. Bakit daw hindi namin naririnig yung mag-test dati? Sabi ko kasi yung mag ginagamit lang natin sa kape. Pero hindi pa tayo gumawa ng mag-test.
Saan narinig yung mag-test?
Hindi ko siya pinapansin sa Enterovac kasi Invic lang. Kasi Invic lang ang ginagamit natin. Game, bakit si Escherichia coli, si EHEC?
It produces the enzyme beta-D-glucuronidase, which hydrolyzes beta-D-glucopyranidase.
Tignan mo, sabi ko sa'yo, ako din nabubulol dyan sa mga yan. Kaya, si methyl amberliferyl. Okay. Sino nagproproduce kay beta-D-glucuronidase? At sila ang ginagamit dyan sa ating MAG test. Andiyan na nga. Andiyan. Kaya nga sila ginagamit for our E. coli and our Enterobacteriaceae. Kasi sinahydrolyze ni Enterobacteriaceae si beta-D-glucopyranidase uronic derivative to aglycons and D-glucuronic acid. Yung substrate natin dyan is your for methyl amberliferyl beta-D-glucuronide or in short, si MAG. Hindi lang pala pang kape, sabi namin ni Arlene. And a fluorogenic substrate of beta-glucuronidase which is impregnated in the disc. Huwag niyong anuhin yung sarili niyo. Bakit test tube yan? Kasi pwedeng dehydrated tube, dry tube, pero andun yung reagent. Pwede rin disc. Okay, so that lalagay mo lang dito yung parang i-drop mo yung inoculum, kakaroon ng reaction. Walang laman. Dehydrated tubes yan or gagamit tayo ng impregnated disc. So that if the organism is capable of producing yung enzyme natin which is your beta-D-glucuronidase, which has the ability to cleave. Ano na ulit pag sinasabi natin yung cleave lagi sa molecular biology? Cleave. Able to cleave. Cleavage daw, sabi ni Ma'am Arlene. Wala ako nun. Ang bad ni Ma'am Arlene. Rado ako cleavage. O, cleave. Ano yung ibig sabihin pag sinabi natin to cleave? Has the ability that your beta-D-glucuronidase enzyme has the ability to cleave the substrate. Ayoko nang i-pronounce, MAG. Ating MAG. Para mag-produce siya ng fluorescent moiety, natanggal na ngayon si G4-methyl amblyferyl na lang siya. Tinanggal niya na si glucuronide because of the ability of your enzyme to cleave or to split the substrate. Okay? So that the end product of hydrolysis already is your 4-methyl amblyferyl, which now fluoresces blue under long wavelength ultraviolet light. Okay? So ano ang kapareho niyang test? Kaya lang yung kanina, ang result niya ay clearing, hindi fluorescence. Nasaan tayo dun sa clearing?
DNase test.
Kinase test. Okay. So, ganun lang ang ano. Yung isa clearing, ito ay fluorescence. May limitations si ating MAG test. Ano ang limitations ni MAG test? Parang kanina sa coagulase, sabi natin, huwag tayong kukuha sa BAP. Bakit hindi tayo kukuha sa BAP? Kasi si blood ay pwede rin, meron siyang pseudoperoxidase activity. So that with our MAG test, hindi rin tayo pwede kumuha ng colony sa mga media na meron silang mga dyes. Like, for example, anong mga media ang matandaan nyo na may dye? Inaantok na si Ma'am Arlene. Siya muna kaya pag lecturing ko para hindi siya antukin.
Ay, naku. Awayin lang nila ako yan.
Awayin nyo daw si Ma'am Arlene pag siya nag-lecture. Dexter.
Ay, lalo na si Dexter.
Dexter. Wala na si Dexter.
Natulog.
Tulog na. Busy na mag-run si Dexter. Ano limitations ng ating mag-test? Ayun, meron. Nag-choppy po kasi, ma'am. Nag-choppy? Sino si Ma'am Arlene? O, game na. Anong oras na ba? Anong nga limitations na? Sabi niya, hindi tayo pwedeng kumuha ng colony sa mga media na may dye. Anong matandaan yung media na merong dye? Ano ang tatanungin ko sana si Rangel. EMB media, ma'am. Okay. Ano pa? MAC. Okay. ENB and MAC. Okay. Now, we only have to perform now our MAG test on oxidase positive organisms. Why? Because some oxidase negative organisms naturally fluoresce. So, hindi na natin kailangan gawan ng MAG test yan. Pag yan ay oxidase negative. So, makikita nyo naman yan pag gumawa kayo ng algorithm ninyo from the very top, pag yan ay gram-negative sa negative catalase yan, so that alam nyo na kung ano yung mga isusunod ninyo. Sino bang gagawin nyo? Remember that all your enterovac are oxidase negative. So, hindi mo ito gagawin sa hindi ka mag-test pag nag-true or false tayo, dapat maintindihan nyo. Ha? So that this one would only for those that are oxidase positive organism. So, kanina meron tayong modified oxidase test. Ngayon meron naman tayong si oxidase test now using our COVAX methods. Ano naman itong si COVAX method? Pero kung titignan nyo, ang kanilang reagent naman mamaya, pareho lang naman. You have still your tetramethyl paraphenyl diamine, which will now in the presence of your enzyme. Anong enzyme natin pag sa ating oxidase test? The presence of oxidase test. Na-mention natin kanina dun sa ating modified oxidase test or our microdase test. Na pag, okay, cytochrome. Thank you, Israel. Thank you, Brandon. So that our oxidase test is used again to identify bacteria that produce, yes, Nides, thank you, cytochrome oxidase, that produce your cytochrome C oxidase, an enzyme of the bacteria electron transport chain. So that when it is present, it oxidizes again the reagent, tetramethyl paraphenyldramine, to indophenols, which is purple in color. So that when the enzyme is not present, the reagent remains reduced and is colorless. Okay. So, twice na na-mention yung ating, yung ating reagent. So, with oxidase test, meron tayong mga kailangan na laging tatandaan. Hindi tayo pwedeng gumamit ng nickel-based alloy wires na pagkuha ng ating colonet ilalagay natin dun sa ating reagent. Bakit? Bakit hindi tayo pwedeng gumamit ng nickel-based alloy wires which contain chromium and iron or yung nichrome na sinasabi natin? Because this would produce false positive reaction. So, pag nag-pick tayo ng colony natin, dapat ang gagamitin natin ay platinum wire or wooden stick. So that medically significant oxidase positive organisms are our Neisseria, Aeromonas, Pseudomonas, Campylobacter, and Pasteurella. So, meron din tayong alternative reagent aside from what has been mentioned, yung ating tetramethyl P, phenylene diamine, pwede tayong gumamit ng para amino dimethyl aniline oxalate, although this is less sensitive, but it is cheaper and more stable than the tetramethyl. Okay? Okay pa kayo? Oxidase test? Anong ating reagent? Reagent ng ating oxidase test? Enzyme yun. That is the presence of the enzyme.
Tetramethyl P-phenyl.
Kaya yan. Sige, tetramethyl.
Paraphenylene diamine po, ma'am.
Yes. Dihydrochloride. Okay? Positive result? Positive result.
Dark purple color po.
Due
Within
to
10
the
seconds.
formation of
Indophenol, ma'am.
endophenol. Okay, pwede na kayo mag-quiz mamaya. So we have the oxidase test for Neisseria wherein our reagent would be our 1% dimethyl p-phenylene diamine hydrochloride. Whereas for the endophenol method, ang ating reagent ay para amino dimethyl aniline hydrochloride or oxalate and alpha naphthol, which will now give us an intense blue color within 2 minutes. Okay, PYR. Robert. Si Robert, nag-ano ko agad siya ng TMPP. Si Mion. Robert, good afternoon daw, sabi ni Ma'am Arlene. PYR test, Robert. Robert. Dave na lang, nawala si Robert. Robert, wala ka mic? Ma'am, di gaan na mic ko. Okay.
Dito lang ako, hindi na gumana.
Dok, lagot ka, Robert. Andito lang daw si Ma'am Arlene, di na gumana mic mo. Dave! Dave! Si Dave Ikin.
Kasi si Ikin,
Dave daw, o sabi ni Ma'am Arlene. PYR. Hindi rin gumana ang mic ni Dave.
Nawala. Pag andito pala ako, hindi gumagana yung mga mic nila.
Dave. O si Nides na lang. Nides. Huwag na daw si Brandon.
Pasawa.
Asa na kayo? Shane. Oxidase. PYR test. Tam na lang si Tam. Tam. Baka natulog na si Tam. Wala na daw kayo, sabi ni Ma'am Arlene. Tam.
Ma'am, ano po yun?
Tingnan mo, natutulog ka na, Tam.
Ma'am, hindi po.
Oh, ang tagal mo sumagot.
Hindi, ma'am.
PYR.
Hi, ma'am. Ito po.
Ayan.
Hindi yan.
Ma'am, it is used for the identification of Streptococcus pyogenes and Enterococci by the presence of... Yun, ma'am.
Ano yun? Ano yun?
Ano yung L?
Ha?
Ma'am, basta yun po.
Ano nga yun? Ah,
Ma'am,
Okay,
binabash
si Ma'am
kasi
Arlene
ako ni
sa'yo.
Dexter.
Ay, Dexter ah.
Dexter, wag mo daw ibabash si Tam, sabi ni Ma'am Arlene. Game na, Tam. Ano yung enzyme mo? We're still on enzyme test. So, anong
Tapos,
enzyme mo? Ano ang mag-hydrolyze ng iyong L-pyroglutamic acid beta na
laboratory.
impregnated into the test disc? Tam, saan ka na?
Ma'am, di ko get yung reaction.
Di mo get yung reaction. Di ba sabi natin, pag nasa enzyme tayo, ano ang role ni enzyme
Po,
natin?
ma'am.
Ah? So that we say now, di ba sabi natin dyan, it is
Yes.
presumptive identification of group A beta hemolytic streptococci.
Ma'am,
Ayan, streptococcus
Natalie.
parang. Okay, and enterococci based on the activity of the enzyme pyrrolidonyl acrylamidase. Si pyrrolidonyl acrylamidase or our PYR, also known as our pyrrolidonyl aminopeptidase, is a bacterial enzyme that hydrolyzes now our L-pyroglutamic acid beta-naphthylamide impregnated into the test disc. So nakita nyo parang lahat ng mga disc kanina that serves now as the substrate for the detection of our enzyme. Ano si enzyme natin? Our pyrrolidonyl acrylamidase. So, yung hydrolysis ng substrates will now yield yung ating beta-naphthylamine, kita mo yun, plus, sabi niya, which combines with the PYR reagent. Ano si PYR reagent? Is our para-dimethyl aminosinamaldehyde to form now our bright pink to cherry red. So, bright pink to cherry red color. So, this test can also be performed either pwede sa broth, pwede rin siya ngayon sa paper disc. Pero what is being shown to you here is the paper disc test. Okay? So, again, you come up here with ano ang action ni enzyme doon sa substrate. Kaya yung nakikita nyo dyan na L-pyrrolidonyl beta-naphthylamide, ano mangyayari sa kanya at magiging beta-naphthylamine siya, magre-react siya with the reagent methyl aminosinamaldehyde, which will now give us the bright red precipitate o pwedeng bright pink to cherry red color. Okay? So, the bright red should be there again. Take note of the time of incubation. It should be within 5 minutes after the addition of the reagent. Okay? So, Yun yung mga enzymes natin. If I'm going to ask you to recall, what are the different enzymes that we have discussed? Starting from ano yung unang enzyme natin? We had our butyrate esterase. That is what we use with our butyrate this test. And then you have your catalase test. So that is also your catalase enzyme, giving us now what? Bubbling due to the formation or the breakdown of H2O2 into water and oxygen. Then you have your coagulase test. Of course, free or bound coagulation. Our positive result is still clumping or the formation of clot. And we have our DNA hydrolysis. Ano yung ating DNA hydrolysis? It is the clearing around the colony. Hindi pwedeng clearing lang ha. It's clearing around the colony. And gelatin hydrolysis, anong ating positive result? In the presence of the enzyme gelatinase is partial or total liquefaction. And then, sa ating hipurate test, anong ating positive result? Due to the formation of what? We have now our deep purple. Okay? Sa ating leucine aminopeptidase, anong ating positive result? This again, red. Bakit nawala si Dexter o bumalik si Dexter? Humina ang iyong net. You will get the red within 1 minute after the addition of cinnamaldehyde and of course your modified oxidase test or your microdase. Again, the presence of our cytochrome C oxidase. Then your MAG test. Ano si MAG? You have your beta-D glucuronidase, which is our enzyme. And of course, again, your COVAX method. So it's the same with your microdase test or your modified oxidase test, wherein the enzyme that is present should be our cytochrome C oxidase. And for PYR, you have your pyrrolidonyl arylamidase. And that is the last of our enzyme test. So, pag may enzyme, ano lagi ang ating reaction? Is either i-break down niya, split, kaya nga andun lagi yung word na cleave, or in the hydrolysis of the substrate. So we proceed into our tolerance test. Sabi natin kanina, anong ibig sabihin pag we're talking about the different tolerance test? Ano ang una niyong maisip pag sinabi natin halophilic ang ating organism? Ano ang tinotolerate ng mga halophilic organisms?
Salt po, ma'am. Increased salt.
Yes, so high salt concentration. Mag-grow pa rin sila kahit ayan ay high salt concentration. So that with our tolerance test, we have our bile osculin test, we have our cetrimide test, we have our salt tolerance test. Ano si bile osculin test? So that our bile osculin test is used to differentiate our enterococci and group D streptococci from the non-group D viridans group streptococci on the basis of their ability now to hydrolyze esculin. Okay? So that the basis of the esculin test is the hydrolysis of esculin, which is a glucoside, in the presence of bile salts as a result of the enzymatic action of esculin.